Brenner and Crick determined that a codon consisted of 3 non-overlapping nucleotides in sequence in order to correspond to all 20 amino acids. The code had to be 3 nucleotides because 2 didn't generate enough codons to code for each amino acid. But how do we know which codon corresponds to which amino acid? Nirenberg and Matthaei conducted an experiment to try to answer this question. They took a poly-U sequence of mRNA, which is a string of consecutive uracil nucleotides, and combined it with a cell-free extract, which includes the components necessary for translation following cell lysis. All 20 amino acids were added to the test tube, with one being radioactively labeled. This was repeated 20 times, resulting in 20 test tubes, each with a different radioactively labeled amino acid. This was done in order to see which radioactively labeled amino acid ends up in the translated protein. The protein couldn't form unless the correct amino acid matching with the UUU codon was incorporated. This showed that the UUU codon specifically codes for phenylalanine. They conducted the same experiment with the other homopolymers possible: poly C sequence, the poly A sequence, and the poly G sequence. They found that the poly C sequence coded for proline, the poly A sequence coded for lysine, and the poly G sequence did not code for any amino acid and didn't work. This determines which amino acids 3 of the 64 codons code for, but what about the other 61? 3 years later, Nirenberg worked with a different scientist named Leder to attempt to determine the remaining 61 codons. In order to do this, they combined a specific trinucleotide such as GUU with a ribosome and 20 charged tRNAs. A charged tRNA has an amino acid bound. One of these charged tRNAs has a radioactive amino acid bound. Time was provided to allow for binding to occur between a charged tRNA, the ribosome, and the trinucleotide. The mixture was then passed through a nitrocellulose filter, which prevented tRNAs bound to the ribosome and trinucleotides from going through. The filter was then analyzed to determine which amino acid was bound to the tRNA. The filter was also tested for radioactivity. If radioactive, then the trinucleotide codes for the radioactive amino acid. This process was repeated with the same setup, but by substituting a different trinucleotide to determine which amino acid it is associated with. Using this method, Nirenberg and Leder were able to determine which amino acid is associated with the remaining 61 codons.